design and control of bioprocesses. Before I go to the main topic, let me give you a brief overview of what is happening in the field of industrial biotechnology. So there are about 400 different kinds of firms or companies in this field, with an annual turnover of about 8,000 crores. We can broadly divide these companies into healthcare, food and agriculture, and industrial biotechnology. And as you can see, the dominant impact is in the field of healthcare and natural resources. Now, if you look at the activity, what are different types of things happening in the field of biotechnology, you will see in this pie chart that the dominant activity is in academics. There is a lot of research going on in traditional biosciences departments as well as several engineering departments now, followed by human health. I'm going to today talk about one application in the area of human health, and that deals with our effort of producing therapeutic proteins using a microorganism Bioprocesses. Pikia is considered as an emerging star for making a host of therapeutics. Now, if you look at the global therapeutic protein market, it's currently at around $100 billion US, and it's expected to rise to about 150 by year 2070. There are various proteins which have been approved by FDA, and Bioprocesses is one of the organisms which is playing an important role here. At present, majority of these therapeutic proteins or the proteins that we need for healthcare are produced by the mammalian cell line or a bacteria E. coli. But Pikia offers a distinct advantage in being an yeast which has a glycosylation pathway which is present in human beings. Now, I'll come back to this example of Pikia a little later, but let's try to understand when we say we are going to carry out an bioprocess, what exactly is happening in this process. So, if you like, we can say that there is a multi-scale view of a bioprocess which you see on the screen there. So, on one side, we have a traditional bioreactor, our good third tank reactor, and in which we are supplying various different nutrients. If you are operating it in a continuous mode, we will take out the product continuously. We have our aeration, agitation, and the usual stuff. What distinguishes the normal chemical operation from a biological operation or a bioprocess is presence of these tiny microorganisms, which we expand here is the workhorse of our bioprocess. And what happens inside the microorganism is what makes it very interesting. So, for example, we have a lot of information organized inside the cell, starting with the genome, which contains all the necessary required information to carry out various processes. Now, the reason we are using this microorganism is to get a desired product, and all the information that is required for these cells to make the desired product is contained in this genome. However, the journey from a genotype, that is information in the genome, to a phenotype, namely the ability of microorganisms to make different proteins, goes through several steps. For example, we go from genetic information to through messenger RNA to proteomic information, and these proteins in turn drive variety of metabolic reactions, and these metabolic reactions in turn are responsible for producing the product of business. Now, if you look at this as a reaction engineering problem, all these genome to proteome to metabolome are variety of complex reaction networks, and these networks are interregulated. So there is a lot of feedback from for example, from a metabolic step onto the proteomic step and a genomic step, and therefore this becomes a very highly regulated environment. Now, in the reactor, we are providing the essential nutrients, which in turn influences regulation of this network, and thereby giving us an opportunity to control these reactors in a manner in which we get the best from the cell. So keeping this in mind, let us try to see what is the effect of all these different regulatory networks and their control on the observed behavior of the cell. And this is what makes these biological processes rather unique. The thing that comes to mind is the environmental regulation of the rate and mechanism of the reaction. Now, because it is a tightly controlled network, the optimal conditions that are required are often within a very narrow range. For example, the substrate concentration or the carbon source is inhibitory if it exceeds, for example, a gram per liter, or the cells are deficient if it is below 0.5 grams per liter. So one can say that the range in which we have to operate now is restricted to between 0.5 to 1. Same story repeats for variety of different other constituents of our liquid media, as well as the physical conditions such as temperature and pH. In fact, cells are extremely sensitive to variations in both these parameters. For example, certain type of cells can tolerate only up to temperature in the range of 25 to 30 Fahrenheit, or pH between 4 to 5. Any deviation both below and above this condition will lead to loss of activity by the cell, and thereby meeting this bioprocess to be useless as far as the production is. Now, for cells, bioreactor is a very unusual element because they have not developed themselves over millions of years, so that in 20th century or 21st century, somebody is going to put this cell in a bioreactor. Or in other words, cells in general survive a very harsh condition and often adapt themselves so that they can carry out the function in the environment in which they find themselves. Or in other words, contained in that regulatory network is also ability to adapt themselves to variety of different conditions. And what happens when you put these cells into bioreactors is cells continuously try to adapt, and thereby leading to either variations in the selectivity of a desired product or how much of it we can make, or for some reason If suboptimal conditions exist in the reactor, then these actually could undergo mutations to cope up with these suboptimal conditions. Now, these mutations or these adaptations can occur within few hours, or it could take few months or even years. Typically, when we talk about bioreactors, we are looking at the time scales of hours to weeks at best. Now, another unique aspect of biological processes is the presence of multi-phase gas liquid solid systems. Gas, because
Yeah.
cells, most of the cells, just as we do, require oxygen, and therefore you have to continuously keep on bubbling air. So liquid, because for majority of these processes, the nutrients come from the liquid media. And the solids, although solids is not an appropriate term, the cells themselves present an distinct phase. It's a phase which separates one liquid environment from another through a membrane or cell wall, making this a truly multi-phase system. Now, as far as this regulation inside the cell, the control of the cell often lends itself to a very complicated and sometimes difficult to control dynamic behavior. For example, you may have a situation where cells may suddenly change from one steady state to another steady state, which may or may not be desirable. So all these aspects make biological process interesting both from optimization and control point of view. So now let's come to optimal control of bioprocesses. Now, when we say we want to design a bioprocess, what exactly are we trying to do? What we are trying to do is channelize the metabolic capacity of a selected cell to the production of desired product to maximum extent possible. So we are taking the metabolic capability of the cell and now through environmental changes or otherwise try to channelize this capacity to make a desired product. So how do we do this channeling? Now, this channeling can be done in a variety of different ways. At a more fundamental way or a molecular level, we can alter the metabolism of inside the cell. This amounts to changing the reaction structure or the network structure inside the cell by doing something what we call recombinant DNA technology. So that's one way of doing it, and this has become from art. It is now becoming an precise science because we know exactly how this metabolic network is being controlled by the information in the genome of the cell. So if we can change that genomic information, then we can alter the metabolism of the cell. The more traditional way of achieving this optimal channeling is to do by controlling the nutritional and environment outside the cell, which in turn influences what is happening inside the cell. And this is where I will be focusing, my example will focus later in this talk. Now, when we say we want to optimize and control, what exactly are we looking for? So, given a reactor, let's say that a semi-batch reactor, I'm looking for the best operating strategy to carry out this fermentation process or a bioprocess. So essentially, we are searching for the best solution among various alternatives to maximize the production and efficiency. Now, control, for example, on the other hand, is required to improve and maintain the product quality, detect and eliminate the disturbances. Now, if you look at the last bullet point, the second bullet point on the slide, optimization and control is something which is not alien to chemical engineering. We have been doing this for at least more than 5-6 decades through a formalized strategy for chemical processes. What is different is the first bullet point, namely the bioprocess, where we are trying to deal with a living cell to do exactly what same thing that we have been doing for chemical processes. So let's go back and focus on what are the requirements for an optimal control. So given these objectives, what is it that we require? So we start with, we require a subjective function. So we need a function or something which needs to be maximized or minimized or a combination of these two. For example, a typical criteria is maximize the profit. I mean, that's the most natural thing that comes to mind. But while maximizing the profit, what is it that we are looking for? We are looking for making maximum amount of the protein at the lowest cost possible. Now, in order to achieve this objective, how do we go about doing that? And it is here where we can look at the whole system in terms of various variables that we have to deal with. For example, we can broadly classify these variables, and there are different ways of doing this. But one such way is to say that we have decision variables. That is, these are the variables whom we need to find out so that we get our desired objective. On the other hand, whatever decisions that we take in turn determine the status of the bioprocess or a fermentation process, which we can represent using what we call state variables. So nothing but variables which represent the status of the process. So simply it can be the concentrations of the carbon source, nitrogen source, oxygen, and so on and so forth. Now, any optimization is meaningless or doesn't even exist if there are no constraints. If there are no constraints, then there is no optimization problem. And so there are constraints which restrict the behavior of these variables. The interaction between the decision variable and the state variable and the objective function, and so on. So the constraints become an important constituent of an optimal design. So let's try to address what are the typical constraints on biological processes. So these can be broadly classified into a variety of 4 different types. For example, physicochemical constraints. However fancy or exotic these living cells may look, they cannot violate the basic mass and energy balance. So the same thing that we learn in our chemical process calculations, doing all kinds of balances, have to be applied and cells have to satisfy. Or in other words, I cannot hope to produce 10 grams of cells by providing only 1 gram of mass, total mass, that is either as a carbon source or whatever. So if we supply 1 gram of mass, we cannot convert that in more than 1 gram. It's substantially low. In fact, we can convert it only to about 10% of the biomass or cell mass. But then, so these are common concepts. that arise in any process, let alone bioprocess. But what makes biological processes interesting are the remaining constraints, such as biological constraints. So we may provide a carbon source, let's say glucose, and hope that cells converts it into an product such as insulin. However, for glucose to get converted into insulin, the whole carbon has to go through what's called metabolic pathway or metabolic network. And it's not a single step reaction. There are hundreds of reactions involving hundreds of different biomolecules. Now, these reactions in turn are controlled by or catalyzed by enzymes. Enzymes are nothing but biological factors. And therefore, in order that these enzymes control these networks, we have to, so conversion of this glucose to insulin, even though the network may be present, it's still regulated by how active or otherwise these enzymes are. Then we have these environmental constraints, namely nutrient ability, availability, pH, temperature, and so on. In addition to all these constraints mentioned, there are something called regulatory constraints. For example, these enzymes which I mentioned earlier, which are the catalysts, are subject to activations and inhibitions. So this is how cells will control whether it sends glucose to insulin or not. Now, so the control in when we say activation and inhibition, the enzyme is physically present inside the cell, but it's either in the activated state or an inhibited state. On the other hand, there is also active control by making sure whether the enzyme presents itself or not. And this is the control exerted at the genomic level or genetic level, what we call the gene expression. So let me give you few general examples, and these examples are selected at random, so there is no preference on it. For example, one will be optimally design a strain. The objective here is to maximize, let's say, certain growth or metabolic product of interest. The decision variables or decisions that we have to make are how to alter my genetic network. So what genes do I add and what genes do I delete so that this channeling is achieved. Now, the constraint specifically in this particular case is that I cannot arbitrarily go on deleting any gene, a gene which is absolutely essential for the survival of the cell. So the constraint, for example, I can do this manipulation as long as my metabolic network inside the cell is perfectly stoichiometrically balanced. Second example is let's say about media design. So how do I design or how do I provide the required nutrients to achieve the same growth or maximize the products and so on. So decision variables here are media constituents, the pH, the temperature, and the constraint of course is again the microorganism's ability to withstand the variations in pH, temperature, and media, and so on. Now, continuing along more examples, we can have fed-batch fermentation, and this is something that I'm going to talk about a little later on. and therefore I will not dwell on this. But another example for instance is designing a multi-production plant. Now, what happens for bioprocesses is that depending on the type of product that we are handling, for example, let's say we are dealing with alcohol, then alcohol is required in huge quantity. So this becomes a classical home project or final year project of design 10,000 tons per annum alcohol production or fermentation facility. On the other hand, if you are designing a therapeutic protein which is required in extremely small quantity, for example, some of the interleukins and interferons, the worldwide consumption of this product may be less than a kg. The entire world requires only about a kg. And therefore, dedicating an entire process plant to make 1 kg of a product in an annual cycle is of course not desirable. So what we are looking therefore is to make multiple products from the same plant, and therefore the problem of multi-production facility. And then you have normal what kind of plant we need, what kind of individual process variables, and so on. Let me give you a brief idea of what kind of problems that arise when we are doing bioprocess optimization, in particular reactor optimization. So, first thing that normally one is confronted with is what kind of reactor or how do I operate my reactor? So I said it's a continuous, a third type reactor. Do I operate it as a batch reactor? Do I operate it as a semi-batch reactor? Or do I do it as a continuous process? So one can actually, we have done this trying to use the optimization tool to determine for a given process what kind of reactor will give me the best performance. Is it batch, semi-batch, or fed-batch? In the biological terms, semi-batch reactor is also called as fed-batch reactor or continuous. Now, the other normal requirement is, of course, we want to get better products at lower cost and so on. So we need to improve the process. So we can use optimization tools to achieve this. And this is something that I will illustrate with an example following the next slide. Now, one of the things in biological process which makes it difficult is we don't have good models. Our models or our understanding of the cell and quantification of that is rather limited, and therefore we have no model. Now, how do we optimize a process if we don't have model? So what we can do is use of measurements. Now, fortunately, fermentation processes we take lot of measurements from temperature case to off gas to some liquid nutrients and so on. So one can actually use a black box neural network approach to do the optimization. Even if we have measurements, sometimes these measurements are fewer in number and as well as not available continuously. So how do we combine offline measurements with online measurements to achieve the best objective? And finally, after doing all this, we actually run the process. Process faces a lot of uncertainty. For example, simple things such as from where do I get my carbon? Now, if you are using a pure carbon source such as glucose, which is rather expensive, then this is not a problem. But on the other hand, if you are using molasses, which comes from sugarcane industry, now depending on who supplies my molasses, whether it comes from a region in Maharashtra or MP, the quality of molasses may change. And if that quality changes, that will influence my process. And therefore, I need to continuously track and identify disturbances or changes and then design my optimal solution. So we have been working on variety of these problems, but now let me focus on a particular problem which has caught our attention in recent years. And this is mainly, I would say, not recent years, for quite some time now. Namely, optimal design of a fed-batch. So as you can see here, the fed-batch bioreactor, first of all, it's a stir-packed reactor, and only difference is I'm going to continuously feed the nutrients and operate this reactor in an otherwise batch mode. So there is no continuous output, only batch operation to which certain nutrients are added. So in designing this fermentation process, my objective is to maximize productivity. So I want to make maximum product, maximum yield, or minimize cost, namely cost which arise from raw materials as well as the time I spend on operating this reactor. So when I want to achieve all these objectives, it becomes a multi-objective problem. Now, I talked about decision variables. So, what are my decision variables for this problem? The decision variable for this problem is the feed. So, at what rate do I add my nutrients? Do I add it very rapidly? Do I add it slowly? Or how do I go about doing this? And of course, there can be temperature, pH, and so on. Now, why do I need this fed-batch bioreactor? Now, if I were to summarize all the complex behavior into a simple, easier to quantify behavior, I can say that the key performance determinants of the process are non-monotonic functions of substrate or regulator. What is non-monotonic function? Let's say on the x-axis, we have certain variables which can be state, product, temperature, pH, and so on. And on y-axis here, I have the response of the cell to variation in these variables. And the typical behavior is as follows. Initially, as the levels of this variable increase, the response becomes better and better. However, beyond a certain critical threshold, the cells actually start responding in a negative manner, or in other words, the response deteriorates. And this is what I meant by non-monotonic function. Monotonic function, the response should have kept on increasing as my variables increase. So this now leads to a situation where I have to control my variable in this narrow range of operating conditions. And for fed-batch reactor, the variable of interest is the substrate concentration or the nutrient concentration. And this is what I am going to therefore control. Now, this region, below this optimal or critical region, is where I am not utilizing the potential of the cell to its maximum, or in other words, I am underfeeding the cells, whereas in this region, I am overfeeding the overfeeding the cell. So by appropriately controlling the substrate concentration, I want to avoid both underfeeding as well as overfeeding, and I am always in this range. Now, if we were to look at purely reaction engineering perspective, we can actually write simple reactions where, for example, there are several of them. I will just go through a few of them. So let's say that we have growth and product are coming from the same pathway. So X here is a cell, S here is a nutrient or the carbon source, and the prime result of providing nutrients to cell is cells multiply, they make more of themselves, and in the process they also make certain products. So by writing this as a single reaction, the product that comes out is essential for cells to grow. Now, there are other examples, and you see few of them listed here, where the growth of the cell and the product formation are decoupled. So, this reaction scheme, for example, the cell only grow. There is no product here. Whereas for product, cells have to go through different metabolic network where it makes more product but very few more cells. And we could have situations where either the rate of the single reaction or rate of these two different reactions are controlled by this nutrient level, namely S, in a manner which I described previously. And this is what makes the design important. Because if we don't control, we can't, for example, get neither cells nor product, or here we may get either cells or either product, but that is unlikely to happen. And there are various other scenarios possible. So when we actually do this fed-batch reactor, we can operate them in variety of different ways. For example, I can determine a priori how should this feed rate change as a function of time. It could be a constant, it could be exponentially increasing, decreasing, and so on. Or I can determine them in an optimal manner using some theory and so on. Even after you do this, one cannot hope to say that I will find this optimal or otherwise the feed rate, implement them on the reactor, and everything will be fine. The reason being, I mentioned a lot of disturbances occur, and so we need the control, or in other words, while designing these strategies, we may think that cells will perform in certain way, but in reality, something else may be happening, and therefore we need to feed it on demand. And therefore, this typically results into fermentation systems being highly computer controlled, and the scheme, if you look at, is nothing different than a traditional feedback control system. So you have a process, you have a host of measurements. These measurements give us a lot of data. The data which I can directly use for control algorithm, or I can do certain manipulations with that data and determine the set points. It goes to my control algorithm. It can be a PID control or an advanced control. It guides what control action needs to be done. We carry out this the signals are sent to the process. Now let me come back to this Pichia pastoris fermentation that I mentioned at the beginning of my talk. So what exactly are we trying to do here? So for sake of our example, my nutrients that I am going to control are these two, glycerol and methanol. So that constitutes my environment. this is my cell. This is my Pichia pastoris. It's an yeast cell, and what it does is, if you provide glycerol, it utilizes that glycerol through some kind of a complex network, and which results into making more of these cells. On the other hand, methanol also can be utilized by these cells, and this utilization is controlled by this particular entity or AOX promoter. So this, for sake of simplicity, is something like a switch. So when methanol is present in the system, this switch gets turned on and allows cells to utilize methanol, convert it into formaldehyde, and then enter into a metabolic network which results into more of cells. Now, this is a normal status of how Pichia pastoris survives. So it can survive with glycerol or methanol. Among these, glycerol gives you lot of cells. Methanol is rather limited, but it in principle can use it. Now, we don't want to grow just Pichia pastoris alone. So we want to make lot of different proteins. So some of these proteins I have mentioned. Incidentally, Pichia pastoris is used for making insulin, for example, and Bioreactor in Bangalore is one of the leading manufacturers using this organism. So these proteins do not exist naturally in Pichia. They are our human proteins. So what we do is we take the gene, for example, the gene which is responsible for this protein, attach this switch to that gene and insert that whole structure inside the Pichia. So now what will happen is, if I turn on this switch, cells will not only use methanol, but in the process, the switch for making this protein will also be turned on, and cells will start making a lot of protein, which will come outside, and we will collect that protein later. Now, what is the problem here? The problem is the following. So, if we take this organism, and there are various different. So let me go back to the previous slide and define the problem more precisely. Our objective is to make maximum of this protein. Okay? This protein is going to be made by these cells by utilizing glycerol and methanol. These are my decision variables. At what rate should I supply glycerol and methanol? Now, why is there optimization problem? The optimization problem is for the following reason. Remember, this is a batch process. So it has a fermenter or the reactor at the maximum capacity. It could be 1,000 liters, 10,000 liters, and so on. Now, if I feed glycerol, remember I am doing a fed batch operation. If I feed glycerol so as to fill that entire reactor, all I will have is lot of these cells but no protein, which is not desirable. On the other hand, if I provide the entire reactor with a lot of methanol, cells use this methanol very inefficiently. So I will have very few cells, and very few cells will make very little protein. And therefore, filling up the reactor either with glycerol alone or with methanol alone is not productive. It will not give me a lot of protein. So in other words, I must strike a balance between providing glycerol to make a lot of cells and then providing methanol so that these lot of cells start making lot of protein and I get the protein in a larger quantity. That's my maximum productivity. Now, there are certain standard protocols which industry follow, and this is one of those protocols given by this company in nitrogen company. So what do? The way it is done is initially provide lot of glycerol and allow the cells to consume that glycerol. So glycerol gets consumed progressively. The cells start growing, so you get lot of cells, and then you start feeding glycerol at certain predetermined rate. So this is my predetermined rate. Here it is a constant value which cells consume efficiently. Notice here, in this particular portion of the operation, there is no protein. My protein of interest is this green one, namely HCG. So now, but I have lot of cells. So once I have these cells, I now start supplying methanol shown in this stepwise step model, step increase of methanol, so that the cells start now, this lot of cells will start making the protein. And indeed, what we see here is cells will start accumulating the protein of interest. So these are the standard protocols. But notice here, one of the things that happens. So if you now like to focus, we can say that from the beginning to this particular time, I am doing only glycerol feeding. Whereas from this time onwards, I am doing methanol feeding. So my optimization problem is what would be the duration of this phase as compared to the duration of this particular phase, which is a production phase. And if you look at the standard protocol, the first thing that you will notice here is as soon as we start adding methanol doesn't start making protein immediately. There is certain lag during which the methanol actually accumulates. It accumulates only almost up to 0.5 grams per liter. And these levels of methanol are inhibitory both to cells, so we see that cells don't even try to grow, as well as to making of protein. So we don't get any protein. So that's where the optimization comes in. So let me quickly go through how do we go about doing this optimization. So again, I will summarize. My objective is to maximize HCG yield in minimum operating time by finding out the feed rate of glycerol, feed rate of methanol, and initial glycerol concentration under the constraint of fermenter capacity, the pumping rate, and the dynamics of the cell growth and protein products. So, in order to address this, we have to first formulate the model, and these are the very few maths or equations that you will see. So bear with me. So these are nothing but mass balances on cells, on glycerol, on methanol, for the protein, the total mass, because remember the total mass inside this reactor is increasing as we are putting fresh mass, and this is something that we accounted for lag. I will get the details of that. So what is then associated with this, of course, are the rates of reaction. So for growth on glycerol, on methanol, on consumption of glycerol, consumption of methanol, and so on. So the first thing that we do is use a lot of experimental data to estimate some of these parameters. Now, once this is done, we have our model for the fermentation process. Then I formulate the optimization problem. So say that I want to maximize the protein amount while minimizing the time, and I'm going to achieve this by finding the appropriate feed rates of glycerol and methanol. These are my constraints. Those dynamic mass balances that I wrote, I can write them in the compact form, and this is my mass balance, fermenter capacity, and pumping capacity equation. Now, this is a dynamic optimization problem, and we formulated with using or we solved it using what we call a genetic optimization algorithm. So, without going into too much detail, what I can say is, if we look at those feed rates, we actually get, for simple problems, feed rate minimum phase, that is feeding at maximum capacity of the maximum capacity, minimum, that is 0, and some intermediate. Now, for a given problem, we don't know what this sequence is. So if I call this phase as 1, this as 2, and this as 3, what I don't know is the pattern. That is, what should be the pattern? Max singular or max singular min, and so on. So this is what I'm going to search for. I don't know when to switch from one pattern to another pattern. So those are the switching times that I'm going to search for, and I don't know this intermediate feed rate. So that is also something that I'm going to search for. So this is my search variable. So in order to find my decision variables of the original optimization problem, namely feed rate, if I can find in a computational algorithm based on this pattern, switching time, and this intermediate feed rate, I will have my complete feeding profile. So we do it, as I said, using genetic algorithm, and I'll skip this. This is basically what a genetic algorithm does. It actually just works on the principle of survival of the fittest. So our optimal solution actually emerges as one of the fifth solutions. Then you actually do all mutations and so on with the numerical mutations. So this is how the solution looks like. So when we have our random solution, initially okay. So what we are seeing here is the operating time on the x-axis and the product produced on the y-axis. And initially, randomly generated solutions give us very little protein. And actually go on refining these solutions, 100 generations, 200 generations, 400 generations. We now see a lot of protein and corresponding operating time. Remember, we are doing here what we call a multi-objective optimization, and here we get what we call Pareto optimal front. What is a Pareto optimal front? So once again, on x-axis, I have a operating time or fermentation time, and on y-axis, I have protein amount, what the protein of interest, in this particular case, SC. So this, in the phase scale of this protein and operating time, this called as Pareto front, gives us the following information. It tells us, for example, if my operating time is 100 hours, then the maximum amount of protein that I can get is this particular quantity. Put it other way around, if this is the amount of protein that I desire, then the minimum time that I require is 100 hours. And this is true for all these operating time. For example, one of the things this this tells us that to get any amount of protein, the minimum time required is 40 hours. If your operating available to is only 40 hours or less, forget about making this product. You go for some other product. And as we go on increasing the operating time, the corresponding amount of proteins obtained is more. But further, it also tells us that beyond the critical, let's say, operating time of about 120 hours, no matter what you do, you can operate your reactor at 140, 160, you are not going to get more protein. And this results from other constraints on the process, how much sugar or how much carbon source you can take. Or in other words, the window of operating time available to you is, let's say, from 40 hours to 120 hours. And these are the amount of proteins that you will get. How does the typical fermentation look like in this solution? So we get two kinds of possible solutions which give you the maximum protein. In one case, what it says is you allow cells to grow for certain amount of time. So this is all glycerol phase, and then start supplying methanol. Notice here, both glycerol and methanol are no longer constant. They say that constant rate. These are my optimal rates that I have got, and the resulting protein comes in this production phase, and you get almost 50% higher protein compared to that standard protocol that I mentioned. Now, an alternate possibility is that you grow cells, but grow it for shorter duration and prolong this production phase to a longer, or you provide methanol at a higher rate for a longer duration, and that will also result into substantial amount of protein. In fact, both these policies give almost identical protein. So I'll conclude my talk here. I'm almost approaching my, or rather exceeded my 2 minutes. So these are the green points here that you see are my simulation results. These are some of the experimental results, and you see the few of the experimental results actually follow the optimal predicted quite well. So let me just conclude by saying that advances in molecular biology has created various new avenues for optimal design. We have several challenging problems in other areas such as biomedical research. That is, for example, deciding on, one of my colleagues in my department is working on in treatment of AIDS, what should be the schedule of the dosage, because excess dosage is not good for the patient. So several of these problems can be handled by using some of these tools. I'll be failing in my duty if I don't acknowledge the contributions by my colleagues, namely Rakesh Vijay, in molecular reproduction and developmental genetics department, several of my students over last few years, as well as my sponsors, DSP. I will stop here, and once again, I don't know how the experience has been for the audience, but I enjoyed it delivering lecture. It saved my travel time, and I hope it worked out well. We have a small question answer session, I understand. So please feel free to ask me whatever is on your mind. I'll be happy to answer you. Thank you. Thank you.
questions are, I'll read them out. Dr. Modak, you are able to hear me?
Yeah, please go ahead.
The question is, why is it essential to use a combination of substrate? Why not only methanol?
Okay, in this particular case, uh, the, okay, because cells have ability to uptake both glycerol and methanol, and the molecular switch that we have used is tied to the that AOS promoter which I mentioned. So if I can go back few slides. So the switch that we used here is this AOS. So this particular vehicle for the protein, the gene, was tied to this AOS. So if I give glycerol alone, then this switch won't be turned on, and therefore we will not get any protein. Now there are other switches possible. For example, one could use a gap promoter or a switch which works only on glycerol, but that is not as efficient as this particular switch. And therefore, the common practice is to use both glycerol and switch.
Okay. So we have one more question. This is regarding the Pareto optimal front. The question is, dynamic optimization tools continue to evolve. So which optimization software is being used?
Gaurav, 1 minute. Can you repeat the question?
The question is, in specific context to Pareto dynamic optimization tools continue to evolve. So which optimization software do you use in your research?
Which optimization algorithm? Oh, software. Yeah, many moons ago I was in ICT. We were taught Fortran, and I am stuck with Fortran. So all the codes that I have used are in Fortran. So we used our own code using some of the available genetic algorithm code in Fortran. But that is not an restriction. In fact, incidentally, I can say for scientific computations, Fortran still happens to be one of the good land platforms. We have MATLAB and all kinds of things, but MATLAB is more ease of convenience rather than efficiency of computation. So, for example, we subsequently wrote this program used it in MATLAB, as well as some of my recent students, they came with C, C++, they migrated those algorithms as well. But these, by and large, are our homegrown algorithms and tools.
Okay. So this is the last question which we have. Question is, sir, all the proteins that you mentioned, like HCG, LH, FSH, are synthesized by hypothalamus or thalamus. Is there any methodology to produce them externally by gene expression?
These are all heterodimers. And what was the second part?
Is there any external methodology to produce them by using gene expression? I mean, Can we produce them externally or artificially? The proteins.
I don't know. What do you mean externally? You mean the chemical synthesis?
Yes, can we synthesize them chemically?
Oh, that will be extremely difficult to impossible because these proteins, even in silica, we find that we don't actually get the entire 100% activity that is achieved in a human or mammalian cell. Uh, uh, you see, one of the things which makes these living cells is interesting is the following. What has happened is we now have a lot of information about genes. So one part of the system is known. Then we have information about metabolic network. So another part is so if we now say that cell consists of these multi parts, actually the information of one part plus information of the another part doesn't make complete information of the cell. That is 1 plus 1 doesn't result into 2. There are a lot of unknowns which need still to be filled. And one of them, therefore, is that even if we know the sequence, we know the chemical composition, how do we exactly go on assembling them? And that's what is, I believe, not possible chemically right now.
So these were the questions we students had. Now, our executive controller would like to thank you personally. So,
Professor Modak, can you hear me?
Yeah, I can not only hear you, I can see you also.
Okay, thank you, Jayant. On behalf of Exergy, on behalf of the Institute, I'm thanking you personally as well as, on behalf of everybody. So, we are looking forward to many such interactions, either electronically or still better personally, because you in person is no replacement. Okay, we will agree with that. Jayant, thank you very much.
Thanks. It was a pleasure.
So, please let us know. Sorry, I am not able to hear you.
No, I said it is a pleasure. I don't know how it was received at your end. And I agree. There is no substitution to personal meeting, but this also is okay. I mean, not bad.
Yeah, I think, yeah. Yesterday, Satish Pandekar was here and we remembered you. And today you are here. So I think it's a privilege for us and my batch of 1980 to become in as well. Thank you. Thank you, Jayant.
Thank you. Bye. I will log off now.
Yeah.
You are currently the only person in this conference.